Okay, let's start our discussion of conservation genetics, focusing on taxonomy. So, taxonomy is, in general, the different types of species, identifying species, right? And so, why is species identification important for conservation biology? It's important for a couple of different reasons. I'm going to try to go relatively quickly through this because as I've been putting together these lectures, I have a lot of content and I want to try to make sure I don't overspend the time that you're supposed to be watching on these videos. Although, of course, me explaining all that took more time. Okay, so why is species identification important? It's important for helping to identify biodiversity, which is illustrated here. It's important for helping management practices, so identifying invasive species, things like that. And it's also important for providing insight into the ecology and evolution of different taxa. And one of the things that's mentioned in this portion of the chapter is how eDNA monitoring, which we've talked about previously when we talked about different molecular techniques to identify species, eDNA monitoring can help assess the distributions of what are called species at risk, or SARs for short. So there are a couple reasons why eDNA monitoring in particular is so useful for this. First of all, it's beneficial for species if they have a really small population size, or they're rare, or they're hard to identify for some reason by traditional survey methods where you have to visually find the species. So this example shown here on the left is showing different species of freshwater pearly mussels that have these larvae that are really small and they actually burrow, and so they have these cryptic stages, hiding behavior, and traditional surveys are really hard to actually identify whether these species are there or not. But eDNA is a really good alternative that helps get around all of those issues. Another reason why eDNA monitoring is good for these types of studies is oftentimes if these populations are declining or threatened, there are legal restrictions for handling the species. And so that can cause complications for traditional survey methods like trapping or something like that. What's shown here is a pike net, which is often used in aquatic studies where you're physically collecting and trapping the organisms themselves. But if you're doing eDNA, all you'd need to do instead is take a sample of that water and you'd be able to know whether the species you're interested in is present in this stream or not. A couple just really cool examples of eDNA that are mentioned in your textbook. The one shown here on the left is this little guy is the golden tree frog, which is found in Trinidad. It's endangered. And they've been using eDNA to collect water from the bromeliads where these frogs live. So traditional surveys, they'd actually have to destroy this bromeliad to be able to find and get to the frog. But what they can do now is just take some water that gathered in that bromeliad and they're able to use eDNA to determine whether that species is there or not. Another really cool example that's shown here on the right, and this was actually something I had been wondering about, was whether you can only use eDNA for aquatic species, right? So a lot of what we talk about with eDNA is taking water samples. They've actually found now that when species like coyotes, which are shown here, when they drink water, they actually release some buccal cells into the water, and saliva as well. And so what you can do is actually collect water from a source that an animal like a coyote has been using to drink, and you can detect through eDNA that a coyote has been there. So it's a really cool application of eDNA that we can use even if the species doesn't live in water per se. If they're using that as a drinking hole, we can still detect their eDNA in that manner. Okay, so that's identifying different species. And another thing that's often used in conservation is identifying what are called subspecies. And subspecies differentiate groups that reflect unique evolutionary adaptations and therefore may justify separate conservation plans. So that's why identifying subspecies is important in terms of conservation. We want to know whether they are acting different enough or have different phenotypes or ecologies or some kind of adaptations that make them separate enough where we should be managing them separately. So the picture here on the right are different subspecies of gray wolves. So they're all Canis lupus, but they are different subspecies. So when we're thinking about how to identify subspecies and how this is related, again, to this molecular ecology class, it's really important to note that if you're identifying species based on genetic differences, which are often done using a marker of mitochondrial DNA, versus identifying species based on their phenotypes, which are their physical characteristics like morphology, their coloration, or their ecological characteristics, you often come to different conclusions. So here, this is what's called a graphical abstract from this paper here. Graphical abstracts are something that are used increasingly in the field now, where you have your written abstract and you also have some kind of image that summarizes what your paper is about. And so in this paper, they were making the point that if you're identifying subspecies based on morphology, that is not going to give you the same answer as identifying subspecies based on genetic data, like mitochondrial DNA, for instance. So in this paper, they were really making the case that we should be using multiple lines of evidence to make sure we're distinguishing subspecies as accurately as possible. So if we're moving on down, becoming even more specific now below the level of subspecies, we also have something that are called conservation units. And if this is all confusing to you, I understand that. These were all kind of hard for me to keep track of in my head as well. So a lot of this is just for you to know and be able to recognize, because I think if you're reading papers, you're going to see these terms if you haven't already. So my main goal here is just for you to understand generally what these terms mean and how specifically molecular ecology data can be used to help inform the identification of them. So again, this is below the level of subspecies. And here we have something called conservation units, abbreviated as CUs, that identify populations that are distinct from one another in terms of their ecology, their demography, or their evolutionary history. One common example of a conservation unit is abbreviated an ESU, or evolutionary significant unit. Again, ESU is the abbreviation. And ESUs classify groups of populations that are reproductively isolated, meaning they are not having gene flow with each other, they are not reproducing between these two different units, and because they're reproductively isolated, they may have these unique adaptations and differences. So in this cartoon here in the right, this is a cartoon I like to use for reproductive isolation. If you have some kind of barrier, in this case a river, that may be preventing individuals from these two different populations from reproducing with each other. So ESUs are one type of conservation unit. And again, like we talked about previously, it's really important to think about what type of data we're using to identify these ESUs. And several lines of evidence, several studies have indicated that using genetic data may be more accurate than identifying subspecies based on morphological data. So in this paper that I have cited here, they were looking at the blacklip pearl oyster, which has a distribution that's shown here in gray. Each of these circles is a different sampling site for this study. And based on morphological data, they identified two different groups or subspecies in the Pacific. However, when they used genetic data, they used, I believe it was more than 18,000 genome-wide markers. They used 18,000 SNPs, and they created a phylogenetic tree, which is shown here. They found, based on genetic data, actually no evidence for that differentiation in the Pacific. They instead found that overall the Pacific should be one singular group, and the only possible differentiation they found for evidence for ESUs is shown here, highlighted in blue and in red, and these were groups found in Tanzania and Iran. So Tanzania and Iran. So those are the only genetic differences they found that actually indicated those two populations should be managed separately. And so you can see here on the right, far right here, they're showing the different morphology of these same blacklip pearl oyster species, Pinctada margaritifera, I probably said that wrong. But what you can see is there is morphology difference, right? But if you look at the genetics, that gives us a different picture. And if we are using genetics, this also ties into everything we've talked about previously. We're getting to the end of the semester now where everything is kind of tying back to what we've talked about previously, which is great. When we're thinking about what type of genetic information to use to identify conservation units, it's really best if we're using both neutral and adaptive markers. So remember, neutral markers are ones that don't affect fitness. Adaptive markers are ones like the SNPs in the red regions here that are under selection, that are causing some kind of functional adaptive difference in two different populations. And an example of this, where you can get different answers if you're using neutral versus adaptive data sets, was found in European hake, which is a type of fish. They don't look like that, but I can't draw a hake, so you can pretend that's a hake. And what they did is use different types of markers to try and identify how many different genetic structural groups or genetic populations are present. And here they're using that structure technique we've talked about previously, which is what you can use to identify non-a priori populations. So remember, you're just putting the data in without any preconceptions about how many different populations there are, but you're just letting the data tell you. The top line A here is showing us the population groupings based on neutral markers. So remember, in these structure plots, each vertical bar is an individual. The different names or abbreviations here on the x-axis indicate different geographic sites where those individuals were collected from. And then the height of the colored bar indicates a probability of being assigned to a certain group. So all of these individuals over here have a very high probability of being assigned to that green group. All the individuals over here have a very high probability of being assigned to the red group. So when we create a structure plot based on neutral markers, we see two different groupings. So essentially the one group here, this one's a little funky, but another group here. However, if we use adaptive markers, we actually get a different story. So both B and C here are based on adaptive markers. This is in the Atlantic, and this is in the Mediterranean. And these adaptive markers that they found have functions relating to response to water temperature and salinity, so all things that make sense. And when they break up the markers they're using into adaptive, we start to see more differentiation here. So in the Atlantic, they identified 3 different groups now using adaptive markers. And in the Mediterranean, they also identified 3. And so this is illustrating that when we're using genetic information to identify different conservation units or ESUs, it's important to think about, are we using information that's actually reflecting adaptation to these different habitats? So again, if we just use neutral markers, we would say across all of these populations in the Atlantic and Mediterranean combined, there are only 2 different groups. But if we use outlier loci, adaptive markers that have some kind of functional relationship, we start to see some more fine-scale patterns. And lastly, another example from the textbook that I thought was really interesting and important to keep in mind is that if you are dividing species into conservation units based on neutral markers, you might actually end up leading to the preservation of random genes that aren't adaptive and aren't going to help that particular species. So this was based on a review where they looked at several different small mammals in Australia. These are both small mammals in Australia. I forget the names of them because they were species I had never heard of before, but they're kind of cute. But the idea here is, remember that the process of drift will fix neutral markers that are random, right? So there's no functional advantage to that. That's just by chance you have some markers that are fixed. If you're dividing species based on those neutral markers, you could be unintentionally preserving those random genes, but really what we want to be preserving are these adaptive genes that have these useful functions. So again, overall, when we're identifying these conservation units, it's really important, ideally, to use adaptive markers to give us the most useful type of information that can also help to the best preservation of the populations.